Professor Barry Halliwell, thank you so much for joining us today.
It's a great pleasure.
And thank you also for taking the time to sit down with me a couple of weeks ago, just have a preliminary conversation. It was very interesting. We nearly got carried away because I think we spent about 45 minutes speaking about various subjects that I thought we'd probably best save for the podcast.
Okay.
But to begin, I thought we'd start with a rapid fire round just to introduce the first subject to discuss today, namely ergothioneine, which I certainly consider you to be the world's foremost expert in. What is ergothioneine?
Ergothioneine is a small molecule. It's a bit like the amino acid histidine, but it has a sulfur attached to it. It can only be made by certain fungi, bacteria, and yeasts. Yet, when you take it in the human diet, humans can't make it. But when you take it in the human diet, it's very avidly absorbed and distributed to all the body tissues, retained and reabsorbed by the kidneys, which actually suggests that it's very important to humans. And despite that, it's almost been ignored until about the past 5 to 10 years.
You've answered about 7 of my questions at once there.
Okay.
I should have added some constraints and asked you to try and keep your answers to a sentence or less if it's a rapid fire round, but that's really helpful background and I think tees up the conversation nicely. With respect to what you said, what are relatively typical intakes of ergothioneine if it's made by fungi and therefore present in various foods that we consume? The number is going to vary depending on the population in question. But
Yes.
Based on what we know so far, what are rough intakes for adults?
The biggest dietary source is mushrooms, but ergothioneine is present in meats, in many other plants, because plants take it up from the soil. So if you don't eat many mushrooms, maybe you're taking maybe 5 milligrams a day, and people who eat a lot of mushrooms might be taking something like 15 milligrams a day. There have been attempts to assess population intakes, but the data are very broad.
And which mushrooms have the highest concentrations of ergothioneine?
For the ones that have been measured, I think oyster mushroom and lion's mane, but all mushrooms have a lot of ergothioneine. And there's a big batch-to-batch variation. So the same mushrooms from different suppliers often have different ergothioneine content.
And you spoke about the fact that our bodies avidly take up ergothioneine and retain it, and that the kidneys reabsorb it. Do we think that most or even all tissues in the human body take it up?
We can't actually, of course, examine every tissue in the human body. But in experiments in the laboratory on laboratory animals, we find it in all the tissues. And we certainly find it in things like human brain, where we think it plays an important role.
How much of it do we excrete in urine?
Very little is excreted in urine, in fact, because the kidneys are very efficient at reabsorbing it. Some of it is excreted in feces.
And I think you did some work to examine the amount that's lost in feces. Roughly what quantity are we speaking about each day?
We haven't really studied sufficient samples to give a clear answer. And of course, it may depend on your diet, but it doesn't seem to be a huge amount.
In that work, your suggestion was that up to maybe 300 micrograms might be lost per day on average. Does that sound approximately right?
Based on the small number of samples that we analysed. But we need to analyse a lot more samples. And I suspect if people start taking supplements or they eat a lot of mushrooms, more of it will end up in the feces, because I think the body absorbs it very efficiently, probably up to a point.
Mm-hmm. And do we think that blood levels of ergothioneine are generally related to age?
We studied how blood and plasma ergothioneine change with age. And from about 60 onwards, and this was in a Singaporean population, they slowly decline, but they don't decline to a huge rate.
And is it specifically from about 60 onwards? So in adulthood, are levels relatively stable from, say, age 18 to 60?
That's what we found in our studies. Yes, it's the levels are pretty much, well, I mean, assuming you don't change your diet, because if you start consuming more, the levels will go up. But in people with age, well, let's say healthy people with age, the levels seem more or less constant, and they slowly start to decline from about 60 onwards.
Have we found that levels in whole blood or plasma are related to any diseases or forms of dysfunction?
We showed several years ago that people with mild cognitive impairment have, as compared with age-related normal people, have much lower plasma or blood ergothioneine. And then we went on and we found that subjects with dementia, Alzheimer's disease or vascular dementia, and other groups looked at people with Parkinson's disease. It seems to be people with these age-related diseases have very low plasma or blood ergothioneine. And we recently extended that to elderly people with macular degeneration, a debilitating eye disease. So low ergothioneine seems to be associated with age-related diseases. And in fact, papers from European groups have shown the same thing in elderly people with frailty or cardiovascular disease.
Do we think that the levels of ergothioneine in other tissues generally tend to move in lockstep with the levels in blood?
I can't swear by this in humans because we can't biopsy every human tissue. But we looked at brain material from people with dementia, and levels of ergothioneine are way down. So they're way down in the blood, they're also way down in the brain. Studies on animals, again, show there is a correlation.
There have now been a few preliminary clinical trials on ergothioneine, several of which have been done by yourself and your colleagues. What are some of the highlights from those so far?
Several years ago, we started a double-blind placebo-controlled trial on people with cognitive impairment. And we were seeing if taking ergothioneine would decrease the development of their cognitive impairment. This was a full double-blind placebo-controlled trial. So people were either taking ergothioneine or a placebo, and neither the subjects nor the doctors evaluating them knew who was taking what. And this is the gold standard of clinical trials. Unfortunately, that trial was interrupted by COVID, and we couldn't complete it because it involved people going into the hospital for blood draws and so on. And by the time the hospitals reopened to experimental work, many of the subjects had disappeared or didn't want to continue. So we only had complete data for 19 subjects, but we recently broke the code and we analyzed that, and we analyzed partial data from other subjects. And the first thing that's striking was that, in fact, ergothioneine, as measured by kidney function, blood parameters, liver function, etc., seems to be completely safe in being administered to elderly people for long periods, a year or more. We saw preliminary evidence of improved cognition in the people taking ergothioneine, and preliminary evidence that one of the biomarkers of neurodegeneration, which increased in the subjects on placebo, actually didn't change in the ergothioneine. But just to emphasize, this is a very small number of subjects. There's been another study just published on cognition, and again, a short-term study, and it's identified cognitive improvements. And there are many other studies. In fact, we've just applied for funding in Singapore to redo the whole clinical trial. But we would also add to it a number of the biomarkers of Alzheimer's disease that have been validated in recent years, things that you can measure in the plasma that assess your risk of cognitive impairment and dementia. There's been a huge development of things like phosphorylated tau and serum amyloid and so on. And we plan to build all those into the trial, which we didn't do before.
And how long do we think that ergothioneine has been present on planet Earth?
It seems to be a very ancient molecule because certain anaerobes make it. That's actually quite interesting because everybody thinks of it as an antioxidant, and indeed it does have antioxidant properties, as we and others have shown, but it may well be doing other things as well.
So the fact that it's been around so long and that our bodies take it up and accumulate it and try and hold on to it suggests that it might be quite important to human biology.
Yes, indeed. And in fact, one of the early workers in the field said exactly that in 1959, and nobody took any notice of it. So it's been there for a while, but people have just focused on other antioxidants and they've ignored this one. But of course, I think one of the reasons it's neuroprotective is it's more than just an antioxidant. It has a number of other properties.
You mentioned ergothioneine safety. The European Food Safety Authority has put out some guidelines regarding what it considers to be the upper limit of safe intakes of synthetic ergothioneine. What is that number for adults?
If I remember correctly, and I'd have to go back and check, it's something like 35 milligrams a day. But I'm not sure that's based on, because I'm aware of studies that have not yet been published, not by us, but by another group, whether we're giving 70 or 80 milligrams a day, which seems a lot to me, but they weren't seeing any safety issues. The interesting thing about the European study is they did a rigorous review of the literature, and they even say that ergothioneine can be safely given to pregnant and lactating mothers. In fact, we have evidence that ergothioneine passes from the mother to the child, certainly through breast milk if the child is breastfed, and possibly through the placenta.
And I believe it's typically included in infant formulae, isn't it?
As far as I know, it's not included in infant formulae deliberately, but the basis of many infant formulas is milk or milk powder.
Yeah. Okay.
So we've analyzed a number of formulas, and they do contain ergothioneine. I'm sure that's something that the manufacturers of baby foods are thinking about.
I checked before our conversation, Barry, and I think the EFSA guidelines are 30 milligrams for adults.
Yeah, you may be correct. I couldn't remember the exact figure.
And 20 for kids.
Yeah.
But the point is that those are high quantities.
Those are more than you'd normally get from diet. And you see, this is an interesting thing about ergothioneine. We've identified low blood levels, low plasma levels, as indicators that you're at increased risk of developing age-related diseases. And obviously, you need an intake that gives you a level above that. But we don't know what the optimal intake is. And that's actually true of many nutrients. So the minimum amount of vitamin C you need to prevent scurvy is about maybe 20, 30 milligrams a day. But many RDAs are set higher because they think that extra vitamin E is beneficial. Of course, there is a limit, and we don't know what the limit is for ergothioneine, but many nutrients like vitamin A and vitamin E and iron and zinc are very useful, and sodium, of course, are very useful up to a point. If you start taking huge amounts, they begin to be deleterious. We don't know whether that's true for ergothioneine or not.
That is it for my introductory questions. So feel free to elaborate more from now on. But you've got a very interesting background and an unconventional one. Your PhD was in botany. So how did you end up transitioning to human biology?
Well, it was, I did my bachelor's degree in Oxford, and that's a 4-year degree program. And the final year is mostly a research project. So I ended up doing my research project in the botany department, but it was really on plant biochemistry. And what I ended up finding was that some of the metabolic pathways I was studying were actually catalyzed by hydrogen peroxide. They were non-enzymic reactions. So I got very interested in hydrogen peroxide and free radicals and so on. And this was at a time when very few people were working in that field. So when I got my first academic position at King's College London, I decided to keep on working in this field. I did some more work on plants, and we discovered a cycle that plants use to remove hydrogen peroxide. But then, since I was in a medical school, I switched to working on medical related issues. But of course, if you think about it, plants are very important to us because they provide us with the oxygen we need, and they provide us with a lot of the vitamins we need, especially vitamin C and vitamin E. And of course, people talk a lot about the role of carotenoids and the role of polyphenols like epigallocatechin gallate and so on. And these all come from plants. So I never regretted doing a PhD in botany.
And we'll return to antioxidants later, because without meaning to blow smoke, I think many people consider you to be the world's foremost expert on that subject. And you are the man that has written a book on that subject too. So you're as well placed as anyone to discuss it. You came to ergothioneine in the 1980s, though, is that correct? How did that come about?
Yes, I was working at King's College, and we were developing methods to characterize antioxidants. There was a lot of talk about this being an antioxidant, that being an antioxidant. So we wanted to know how many of these antioxidants actually reacted with biologically relevant oxygen radicals and other reactive oxygen species. From knowing the rate of reaction, you can calculate whether an antioxidant action in vivo is feasible or not at the physiological concentrations, the levels that are present in vivo. So we published a number of papers. So we looked at a whole string of molecules. We looked at taurine, we looked at melatonin, we obviously looked at vitamin C, and we saw some very early work that ergothioneine was an antioxidant. So we looked at that as well. And we characterized its antioxidant properties, and we published a paper on it, I think 1989.
What did you find with respect to taurine? Was it an antioxidant at physiological concentrations?
Taurine itself is a very poor antioxidant, and we published a paper on this. Its precursor, hypotaurine, is actually, in the test tube, much more powerful antioxidant properties. But taurine is actually quite a poor antioxidant. There's been a lot of interest recently on its biological roles and its anti-aging roles, which have turned out to be quite controversial. But what I would say is, if it has beneficial effects, it's probably not by acting as an antioxidant, but by doing something else. I would also say the same about melatonin. Serotonin, the precursor of melatonin, has much more powerful antioxidant properties, again, in test tube experiments.
That's very interesting, because I think a lot of people who have just been listening to people talk online, or maybe reading lay articles about melatonin, would think of that as being a potent antioxidant, and they wouldn't think of serotonin as being one. And I actually hold my hand up here.
In vivo are quite low, so whether it is or not. There's evidence that melatonin itself is not a good antioxidant, but it may act to raise the levels of our own antioxidant defense enzymes.
If people are taking pharmacological doses of melatonin, let's say someone is taking 300 micrograms or more each day, then the plasma concentrations they're going to see are much higher than physiological concentrations. Do you think at those types of levels, it might have some antioxidant effects or some pro-oxidant effects that could in turn raise levels of antioxidant defense enzymes?
Probably even then the levels are too low to do anything, because we measured its rate constants for reaction with biologically important species, and they're actually very low. So you would need millimolar levels, which you never get even in people taking supplements. Again, if it does have antioxidant properties, it's probably influencing endogenous antioxidant systems rather than directly scavenging free radicals itself.
I watched a talk that you shared, and it was put out by the NUS YouTube channel, which for listeners, I will link to. It's a brilliant resource. I really admire the work that you and your colleagues are doing there. In that, one of the things that you discuss is the types of fungi that supply plants with ergothioneine. And obviously, agricultural practices are evolving over time. one of the things that came up in our previous conversation is that hydroponic systems, for instance, might not necessarily supply plants with ergothioneine. Do we think that plant ergothioneine levels are dwindling over time in general, or is it very much context dependent?
Well, I'm pleased to enjoy the NUS series because the National University of Singapore School of Medicine has a regular series on healthy longevity, and they're all available on YouTube for anyone who cares to watch them. And in fact, some of them are actually fascinating, like the recent one by Aubrey de Grey. So we got interested in this issue of plants and ergothioneine when we were measuring in different foodstuffs. And we found that some batches of asparagus were extremely rich in ergothioneine and others weren't. And then we came across a whole literature from agronomists in the United States who said that it's not only humans and other animals who take up ergothioneine, a lot of plants take it up as well. So if your asparagus has been in contact with fungi in the soil, it will absorb ergothioneine. Asparagus can't make ergothioneine. We've never seen a higher plant that can. And there's a scientist called Professor Robert Beelman in the United States, who's looking at the area of modern agricultural practices as disrupting the interactions between fungi and the roots of plants. And that might decrease plant uptake of ergothioneine. But I don't think people have tracked ergothioneine content of crop plants over time. So I think that's something that does need to be done.
And I suppose we could do something similar with animal foods too, if those contain ergothioneine, because their tissues are actively taking it up as well.
Interestingly, animal feed practiced for laboratory animals contains some ergothioneine, but very little. And yet we find ergothioneine in all the animal tissues. So they're obviously taking it up. It's actually also present in some cell culture media because ergothioneine is present in fetal calf serum or other sera which are often added. Some cells, like HeLa cells, contain the ergothioneine transporter, and they will concentrate ergothioneine into themselves, even if there's not very much in the cell culture media. So again, a lot of people don't realize that some cells will have a lot of ergothioneine and others will not.
I imagine this might be one of those neglected experimental variables that historically has fluctuated between labs and contributed to some of the noise and results between different people.
Cell culture is almost a bit like witchcraft, getting cells to grow with adding this thing and that thing. And we have a series of experiments which we put under the heading of artifacts in cell culture, where cell culture studies have given misleading answers to biochemical questions.
Before we get to ergothioneine in humans and its influence on our biology, one of the things that you've noted is that its chemistry is a bit unusual. And obviously our responses to it depend on its chemistry. It can exist in both thiol and thione forms. What's unusual about that, and why does that matter?
It spends most of its time in the thione form. Thiols tend to oxidize and react with a lot of things. Ergothioneine much more stable, which actually is good for us because it's stable in human samples. So you can measure samples that are several years old. It's also fairly stable in food materials. So it's stable to temperature and that kind of thing. Thiones are more stable. So, for example, the major thiol in human cells is glutathione, which is present at high concentrations. And glutathione has many properties. It's an enzyme cofactor, it's an antioxidant, and so on. But being a thiol, it's less stable than ergothioneine. So there's a lot more glutathione in cells than there is ergothioneine, although it depends on the cell type. So again, if you have that much glutathione, how much antioxidant property does ergothioneine add? Which is, of course, one of the questions we've been working on. Or is it doing something else entirely?
Moving on to the pharmacology, if that's the right term, of ergothioneine. When thinking about drugs, people often speak about their absorption, their distribution, their metabolism, their excretion. And we can apply that same framework to nutraceuticals, substances like ergothioneine.
Yeah.
And so starting with the absorption side of things, what's the protein that transports ergothioneine?
The transporter that was actually identified in 2005 by Grundemann's group in Germany is a thing called OCTN1, and it's encoded by the gene SLC22A4. That transporter has been known for a very long time, and it's present in all tissues. But it was only identified in 2005 as an ergothioneine transporter. It can transport other things as well, like acetylcholine, but under physiological conditions, it seems like it's specific for ergothioneine.
Do polymorphisms in that gene, SLC22A4, influence our absorption of ergothioneine?
They may well do. In fact, a number of polymorphisms that affect transport by OCTN1 have been described, but really the Km and the Vmax for ergothioneine have not been well characterized, and it's one of the things that we are doing at the moment. But it has a very high affinity for ergothioneine. Its Km is about 20 micromolar, which is actually very low. So some of the other substrates, the Km values are very much higher and beyond the physiological range. A Vmax is its maximum capacity, how much it can transport. And Km is the concentration at which it's running at half maximum capacity. So it gives you an idea. So 21 micromolar is very low on the range of cellular ergothioneine levels. So it actually means the transporter is very efficient.
Returning to those polymorphisms, there's been some work on adults in Singapore. I understand that some variations in that gene that encodes OCTN1 seem to associate with differences in plasma levels. How big are those differences, do we think?
We don't know. This is something we're in process of doing. We really want to isolate all these different forms of the transporter and actually figure out which are more efficient and which are less efficient. Of course, one interesting thing I mentioned earlier is that people with low levels of ergothioneine are at increased risk of multiple age-related diseases, dementia, Parkinson's disease, macular degeneration, frailty, and so on. One key question is, why do they have low levels? Is it diet? Is it poor absorption, which of course may relate to polymorphisms? Is it that they turn it over faster? That's actually a very important area to assess.
There has been some work knocking out that gene that encodes OCTN1. My understanding is that it doesn't produce very profound phenotypic differences in mice. Is that correct?
That's actually very interesting because if you knock out, there have been two studies, one in Japan and one in our laboratory, where we deleted OCTN1. There is no gross phenotype. The animals look okay. Their lifespan seems to be about the same. They are more prone to a number of toxins. So they seem to be ultra sensitive to things that generate free radicals. We've recently shown that the animals are cognitively impaired. And that's very recent work, which we're in the process of publishing. And also there's been a Japanese paper where they put animals on a completely ergothioneine-free diet and showed the same thing. So you see, normally when you phenotype an animal, you look at its various organs, you look at how long it lives and so on. But it's possible to miss things. And there was a study on the knockout of methionine sulfoxide reductase, which is an antioxidant defense enzyme, where the animals showed no phenotype. But in fact, they do have a phenotype. They can't hear. But normally, how often do you test hearing and sight and so on? Well, we're doing that on the knockout animals. they're definitely cognitively impaired.
And I think that raises a couple of important points. One is what we can think of as being a kind of spotlight or lantern effect, where whatever you're looking at influences the takeaways from the findings. So
Oh, yeah.
maybe there weren't stark phenotypic differences, but if you were looking at different outcomes, then you might have found something. And then the other point is that there's probably an important ergothioneine transport by environment interaction at play. You were speaking about how they're more sensitive to toxins in general. And one thing that we discussed previously, Barry, is that a lot of mice are raised in so-called specific pathogen-free contexts. And so if they were in more pathogen-rich environments, then the differences between those animals with loss of that gene function and their peers with intact OCTN1 activity might be more stark.
Oh, yes, yes. I mean, this is, let's take mice. Mice in animal units are well fed. They are warm. They have very limited exercise, whereas mice in the wild spend most of their time running around looking for something to eat. And they're much more active. And they have problems, especially in cold environments, of maintaining body temperature. So they're very different. Now, you mentioned this term specific pathogen free. It's an interesting one. People often equate it to pathogen free, but it's not the same thing. It means that your animals don't contain certain pathogens. And you can get the same type of mice from different suppliers, which actually host different pathogens. So SPF means they don't have whatever list of pathogens the company specifies, but they can have other things. And certainly this has been reproducibility of things like allergy, infections in mice, allegedly the same mice in different laboratories and from different suppliers, has been a big issue. So some people now say we should be studying mice in the wild. There's even people now studying diseases in pet dogs and cats because they're very heterogeneous and they're in the same environment as their owners. I mean, these are animals developing diseases naturally, not animals being given disease.
At the onset, you noted that fission yeast and some bacteria can synthesize ergothioneine. There are various bacteria that also take up ergothioneine. And I think that you provided some evidence that that's the case. So if we go back to absorption of ergothioneine, is it possible that some of the gut bacteria are taking it up? And why do you think they're doing so?
Well, every animal we've looked at, let's be specific, every mammal we looked at absorbs ergothioneine very well. As I indicated earlier, plants take up ergothioneine extremely well. And many bacteria do exactly the same thing. Again, if you eat a lot of ergothioneine, some of it will go into the feces and it will interact with gut bacteria. And several papers have been published recently on how ergothioneine is metabolized. Now, one of the products of its metabolism is a thing called trimethylamine, which is generated not only from ergothioneine, but also from choline and carnitine, two other important nutrients. And trimethylamine also comes from the diet, and the liver converts it, wherever it comes from, into trimethylamine oxide. And high plasma levels of TMAO are the exact opposite of ergothioneine. They're associated with increased risk of cardiovascular and neurodegenerative disease. So the consensus, although it's not 100%, seems to be that TMAO is something, or high TMAO levels are something you should avoid. We've actually addressed this issue experimentally. We looked at the TMAO levels in subjects being given ergothioneine in the studies we've conducted. And we've also worked with people in our Department of Medicine, Professor Chester Drum, where he's given us a whole load of patient samples, and we've looked for correlations between ergothioneine and TMAO. And our conclusion is that we don't think ergothioneine is a significant source of TMAO. whereas experiments in which they've been giving people choline, for example, can raise TMAO levels. Although, again, choline is a very protective nutrient. It may be you can get a problem if you consume too much of it. That said, some bacteria use ergothioneine to protect themselves. And one classic example was Mycobacterium tuberculosis, which is a well-known human pathogen, very difficult to eradicate. And it makes its own ergothioneine. Whether it also takes it up from the surroundings, we don't actually know. And if you disable that pathway, the organism loses virulence. It actually has two different thiols, ergothioneine and mycophial, and they help protect it against free radicals and other agents generated by our immune system. So again, if people are infected with TB, I'd be cautious about them taking extra ergothioneine. We don't know that tuberculosis takes it up, but we do know that it makes its own and it does use it. Many bacteria take up ergothioneine and they beginning to characterize the transporters. H. pylori does, and it also helps protect H. pylori against oxidants generated by the immune system. Now, of course, when you're talking about the colonic microbiome, that can only deal with ergothioneine that's passing through. Most of it's absorbed in the intestine, unless you're going to take large amounts, and then it passes into the colon. Whereas, of course, H. pylori is in the stomach. So it's conceivably capable of taking up ergothioneine from the diet, which, of course, raises the question of if you have H. pylori infection and you're eating a lot of ergothioneine, are you making things worse? And we don't actually know. The only data I've been able to find is that mushroom consumption is negatively associated with gastric disturbances like gastric cancer. So, the problem with H. pylori is it causes a stomach inflammation, and that inflammation causes gastric dysfunction, and in the worst case scenario, it causes gastric cancer. The solution is to eliminate H. pylori, which can easily be done by a combination of antibiotics. But if it's actually there in your intestine, does consuming a lot of ergothioneine make it worse? Well, we don't know, and I think it's a question well worth answering. What we do know from the data I've been able to dig out from the literature is that consumption of mushrooms is strongly negatively associated with gastritis and gastric cancer. Now, of course, mushrooms contain a whole load of things other than ergothioneine.
It sounds like if uptake of ergothioneine is relatively complete, then even if there are some bacteria within the gut that can take it up, the overall effect on systemic ergothioneine availability is probably quite small.
I would guess it's probably quite small.
And do we know anything about any dietary constituents that can interfere with ergothioneine uptake? We see that with various other things, for example, when things are bound to plant cell walls.
We have not seen any issue, but of course that raises an important point, which is that there are many nutrients in the human diet that seem to be correlated with increased or decreased risk of disease. So if people are thinking about ergothioneine supplements, should they be taking other things as well? And that's an area that really does need exploration. We have not yet found, because the transporter is highly specific, we've not yet found any agents that interfere with ergothioneine uptake.
That specificity is an advantage. I understand that OCTN1 levels in the intestine, at least in mice, fluctuate across a 24-hour day.
Yes, that has been reported.
Yeah, which I suppose might be related to the circadian clock. It might not. That's not necessarily the case. But do you think that there's any reason to think that uptake is going to be quite time of day dependent then?
It may well be. It's just something that has not been studied. I mean, the interesting thing is I worked on ergothioneine, published a paper in 1998 or so, and then stopped working on it. It was only several years ago I went back because I think this molecule's very interesting. And At that time, there were only 3 or 4 papers a year, and it's now going up to maybe 100 papers a year, which is still very small when you see how much is published on polyphenols and carotenoids and vitamin E and so on. So interest is rising, but a lot of fundamental work has simply not been done. Exactly your questions, a whole pharmacokinetic study has not really been done in humans. We studied what happens in young, healthy subjects. We studied what happens in elderly subjects, but we didn't do a complete analysis where you measure fecal levels and all that stuff as well.
And so just on that note, do we know what the elimination half-life is?
The estimated turnover, so that, there is about 3 to 4 weeks. But again, that's pretty much guesswork. The only way you do turnover is properly in humans is to do what they did with vitamin E, which is that they feed deuterated vitamin E's. They watch the levels go up in tissues, then you stop feeding them and you watch the levels come down. You could easily do that with ergothioneine. You'd either use N15 label or deuterated. Expensive experiment.
And that type of labeling can also help us understand where the ergothioneine goes once it's being consumed. So if we return to our pharmacology model, we can speak now about the distribution of ergothioneine between tissues, which I believe is probably quite species dependent. So there's been some work done on mice looking at which tissues have the highest levels, but the relative amounts in different tissues doesn't necessarily map to what we might see in human beings. What do we see in mice, though? Which tissues tend to contain the most ergothioneine?
When you consume ergothioneine, a lot of it goes into the liver in mice. And the level of OCTN1 in human liver is actually reported as much less than mice. There seems to be consensus that it goes into bone marrow in quite high levels, and it goes into all other tissues. We've actually measured ergothioneine levels in human eyes, and there are quite high levels there. It's not only in tissues, it's also in all our body fluids. You find it obviously in plasma, you find it in seminal fluid, you find it in tear fluid. So we've only just begun to study its role in the eye. If you drop ergothioneine solutions onto the eye, it's absorbed because there's OCTN1 in the cornea and it's distributed. Similarly, when you consume ergothioneine, a lot of it gets into the eye.
Would the same thing be true for topical application to the skin?
We have not studied skin, but there was a Japanese paper recently that actually showed that when they fed animals ergothioneine, quite a bit of it ends up in skin. I don't think they've looked at topical application. Ergothioneine is very water soluble, so it's not like it can get across lipid membranes, which is why OCTN1 is so important. But actually, the question of topical application is an interesting one.
I was just thinking if it is accumulating in the skin and then it's thought that it protects the skin against UV damage, then I suppose the cosmetic industry is going to latch onto this at some point.
I think ergothionene's already added to a number of cosmetics.
Is it? Yeah.
Vitamin E is as well, and vitamin C. The other thing worth bearing in mind is a lot of lotions applied to the skin are oil-based, which means that the oils contain double bonds and they're very prone to oxidation. So you need to add antioxidants to preserve your skin creams. And then you can make it a selling point that they're also raising antioxidant levels in the skin.
So could it be then that the ergothioneine in those products is just being used to prevent the oxidation of other substances within them rather than actually ending up being delivered to your body?
Perfectly possible, yes.
You mentioned that levels in bone marrow are very high. Does erythropoiesis generate a lot of oxidative stress?
One interesting thing we find is that when you look at the distribution of ergothioneine in the blood, there's a certain amount in plasma. There's a lot in red blood cells. When we fed ergothioneine to healthy human subjects, and we published this paper a few years ago, the blood levels go up. Now, when we stop feeding, the plasma levels start to come down, but they don't come over the time scale we looked at, which was about a month. They were coming down, but they were not reaching the original level. So in other words, they were still up. The red blood cell levels were still going up. And there've been a number of papers published suggesting that the bone marrow deliberately puts ergothioneine into red blood cells, which would explain why it's still going up, because half-life of red blood cells in humans is often quoted as about 120 days or something like that. So there's a constant supply of new red blood cells. So this actually seemed very interesting. We tried to measure ergothioneine in human bone marrow samples, but it's very difficult to do accurately.
I've seen a report suggesting that by supplementing with ergothioneine, you can increase brain ergothioneine levels by about 5 to 7-fold in mice.
Yes.
Do we have a sense about how much you can boost levels in various tissues by relative to baseline? And does it depend on the baseline? Do we tend to find that tissues with lower levels respond more, or does that seem to be unrelated?
We published a paper on this in mice, and in fact, you can increase brain levels by 5 to 7-fold. Ergothioneine levels increase in every tissue that we examine, so it goes all over the place. Humans, it's very hard to tell. We know that humans with low blood levels of ergothioneine with dementia have low brain levels. But of course, we can't biopsy everybody's brain and spleen and everything and see how much the levels go up. But we do know it gets into human brain. It's even found in the brains of newborns. There was a paper published on that recently of children who very sadly died soon after birth, and they looked at the metabolomics, and the ergothionene's definitely there. So, as I mentioned earlier, it seems to pass from mother to child, certainly through breast milk if there's breastfeeding, and probably also through the placenta. And in fact, there are groups in Scandinavia looking at how ergothioneine levels in children relate to health outcomes, and they've published a number of correlations. But again, you've got to be very careful with correlations.
Do we know for sure that it crosses the blood-brain barrier? Must do.
Well, we know that it's present in the brain, so we presume it crosses the blood-brain barrier. Now, the interesting thing in humans is nobody has ever found OCTN1 in cells of the blood-brain barrier. Now, that could be because getting samples of human brain material is very difficult, and they're often very old, frozen for ages and deteriorated. So it could be that it declines. It could also be that there is a different transporter that takes it into the brain, which again is something we're looking at. There may be other transporters as well. So, for example, when we studied mitochondrial uptake of ergothioneine, if we feed animals ergothioneine, a lot of it goes into mitochondria in brain and liver and other tissues. If we knock out OCTN1, we don't find it in any tissue because it doesn't get through the gut. But if we isolate mitochondria from OCTN1 negative animals, they still take up some ergothioneine, but not as much as ones from animals that do have OCTN1. So again, there may be more than one transporter in mitochondria.
Where else do we find OCTN1 within cells besides mitochondria?
This is something we're looking at. We think it's on definitely on the plasma membrane, of course, but we're looking at endoplasmic reticulum and nuclear membrane and that kind of thing. And this is actually an example of how much fundamental work has not yet been done. So ergothioneine gets into our cells. Where does most of it go? Well, a lot goes into mitochondria, but we've never done detailed cell fractionation. We're in process of doing that.
You've done quite a lot of work on how ergothioneine might be acting within cells. I think most people consider it to be an antioxidant, and it might have some ability to chelate certain metals. So a general theme seems to be protecting delicate structures within cells from oxidative damage. Do you think that it's a good, effective antioxidant at physiological levels?
One of the things we observed was, as I mentioned earlier, in normal cells, there's a lot of glutathione, much more than ergothioneine. So it's hard to imagine antioxidant activity being there in the normal cell. But what we observed is that when you injure tissues, the level of OCTM1 increases and the level of ergothioneine increases. So we were talking about it as an adaptive antioxidant, that you deliberately raise levels. Now, that being said, simple antioxidants like vitamin C, vitamin E have not done that well in improving outcomes of human disease. So what we think about ergothioneine is the antioxidant properties are certainly helpful, but it's also anti-inflammatory. And as you mentioned, it can chelate metal ions like iron, which seem to contribute to Alzheimer's disease and other dementias. Now, there's some very exciting work from Japanese laboratories that suggests that ergothioneine not only promote neurogenesis, but also enhance the actions of neurotrophic factors like BDNF. So this, in fact, will be very exciting. And we're also studying this. It's mainly been done on cell lines and some animal studies. So it really needs pinning down. And we're doing this on human brain organoids at the moment. But if that's true, it's actually quite fascinating, because if it can actually generate new neurons as well as protecting existing ones, that's potentially very important. There's been a recent paper in Cell Metabolism that suggests that it can generate hydrogen sulfide.
Yeah.
And hydrogen sulfide is an interesting molecule. We don't think that's the explanation, because H2S donors don't normally reproduce what ergothioneine does. And hydrogen sulfide is an interesting molecule. A little bit seems to be beneficial, but a large amount is quite clearly toxic. So we've never seen toxicity with ergothioneine. But again, this is a certain possibility. Interestingly, in that issue of Cell Metabolism, there were 2 papers on ergothioneine that actually reached completely different conclusions as to its mechanism of action. So again, a lot more work is needed to be done on this. I honestly believe it has multiple mechanisms of action, and that's why it may well be very neuroprotective.
The other paper, was that the Spiegelman paper?
I can't remember, I don't know, but one said it inhibits a specific enzyme and the other said it doesn't. So this needs to be rationalized. I was actually quite surprised that the editors of Cell Metabolism didn't ask somebody to write a commentary on these 2 papers, which would have been very interesting. But
you should have written a letter to the editor, Barry.
Ergothioneine now in pinning down its mechanism of action.
Going back to the paper by the Japanese scientists, I think they did some lifespan experiments using roundworms, finding that it extended lifespan. They did some work on mice, suggesting that it increased their muscle mass, it improved their endurance in exercise tests, which was associated with increases in angiogenesis. So the system of blood vessels that supply muscles with oxygen and nutrients to support exercise performance. Then mechanistically, they did that work on hydrogen sulfide, suggesting that that was what drove many of those beneficial effects. And it increased levels of NAD, which is a coenzyme that a lot of people in the geroscience world are very interested in. Obviously, now we have NMN and NR and various other nutraceuticals that are being sold for lots of money with the intent of raising cellular NAD levels. What do you think about ergothionine's potential there?
That was actually
a
fascinating observation, because raising NAD levels has been claimed to be beneficial in many studies.
Yeah.
So, as I said, ergothioneine may be doing multiple things. So maybe it does produce some H2S, but not as much as H2S donors, which may be a good thing. Seems to influence NAD levels. I'm not quite sure exactly how, but a mechanism was suggested. Talking about effects on lifespan, we've recently got ergothioneine accepted into the United States Intervention Testing Program, which is a very, very rigorous program, which will look at its effects on lifespan. And they've looked at many of the molecules that have been claimed to increase lifespan, and they found that some work and some don't. So you mentioned supplements, and indeed now you can buy ergothioneine online all over the place, and many companies are getting into manufacturing it. One word of caution, somebody from our Healthy Longevity unit at NUS published a paper recently where they went out and bought NAD precursor supplements, I think nicotinamide riboside, from different health food stores, and they brought them back and analyzed them, and the results were quite shocking, that many of them don't contain the correct amount, or they contain other things, and so on. So this is a problem. That quality control is a big problem in the supplements industry generally, because instead of doing rigorous quality control, you can do a knockoff product and get it onto the market. They were specifically looking at NAD precursors. Again, in ginkgo biloba, there was a study in Hong Kong where they looked at, I think it's 12 or 15 different samples, and they found none of them contained the correct amount of ginkgo biloba. Some of them contained none at all. Again, I always say quality control is absolutely important. The best people working in the field marketing supplements would, I think, generally like more regulation. But certainly in the US, there's very little regulation of supplements.
Yeah, and there has been a lot of that type of research, other work on melatonin, finding that those products contain serotonin, and the list goes on.
Big issue, of course, is there have been multiple studies in human disease on fish oils. And fish oils oxidize like crazy.
Yeah.
And you have to be aware of this, and you have to check for it every so often, every week or every month. And if you look at many of these clinical studies, which were expensive double-blind studies, there was very little, if any, characterization of the state of the fish oil sample. I'm pretty sure some of them were feeding oxidized fish oil and others weren't. And that might explain why there's such an amazing variety of outcomes in these studies.
Being pragmatic for listeners, if you use a fish oil supplement, then you need to store it somewhere cool and somewhere dark. So put it in the fridge, please.
Yes, absolutely. And quite often these supplements will have added antioxidants. The commonest one is vitamin E. But vitamin E by itself without vitamin C is not that good at inhibiting lipid oxidation. Again, you need to, as you say, you need to store these things properly.
The food industry is using lots of others nowadays too. One that I've seen used more frequently to minimize oxidation is rosemary extract. Do you have any thoughts on that?
Yeah, I mean, I had a postdoc in my lab, Professor Arruoma, many years ago, and he was very interested in rosemary because it contains some quite powerful antioxidants. So I presume they've done the correct toxicology on it. But I mean, ginger as well contains antioxidants. So a lot of plants contain antioxidants. So a number of plant extracts have been tested, and they're regarded as more natural than things like butylated hydroxytoluene, DHA, and so on.
Going back to the interventions testing program, the ITP that you mentioned, something occurred to me when you were speaking, and that is that looking across the different candidates that have been tested, most of them extend lifespan in male mice specifically. There aren't many that extend female lifespan. Rapamycin seems to do so in both sexes. Rapamycin combined with a couple of others does. Glycine does similarly in both sexes, but that's basically it when it comes to females. And something that you've noted is that, I think this is true of human females, they have higher endogenous antioxidant defense levels than males do. If one of the mechanisms by which some of those candidates is acting is via antioxidation, be it direct or indirect, then that might explain that sex difference. Do you have any thoughts on that?
Well, it's well known that women live longer than men
Mm.
in terms of lifespan. Health span tends to be a bit bigger as well, but it varies. There's a guy in Spain called Professor Vina, Vina, who's actually looked at antioxidant levels in male and female, I think mice. And he's argued that female sex hormones have a role in raising endogenous antioxidant enzyme levels. So there may be some truth in this. There's also a hypothesis which came from a guy called Professor Sullivan, who's now passed away, that although iron is essential in the human body, too much iron is bad for you because it stimulates free radical reactions. And what he pointed out was body iron levels are lower in women, except after the menopause when they stop menstrual bleeding and then the levels catch up. So he attributed it to lower iron levels. But it's a very clear-cut phenomenon. And it actually illustrates the importance of when you do animal studies to pay attention to a number of issues, and one of which, of course, is the gender of the animals.
One of the subjects that I personally find very interesting, in part because it's practically free, minus the opportunity cost, at least in most countries, is blood donation, which does seem to be associated with some health benefits. And it's self-limiting because if you turn up at the centre to give blood and some proxy of your bodily iron stores is too low, then you won't be able to give blood. So in that way, I'm not too concerned about recommending that people go out and give blood. But it does seem to associate with many different health benefits. And the epidemiology backs it up, and the limited experimental studies support that too. And some of that probably relates to antioxidation. Some of it might also relate to offloading some so-called forever chemicals. There's been some interesting work on firefights in Australia showing that to be the case. Do you have any thoughts on blood donation as an antioxidant intervention?
Epidemiology often relies on correlation. And if you take ergothioneine, low ergothioneine correlates with, and in some cases predicts, increased risk of neurodegeneration, macular degeneration, frailty, and so on. But is this cause-consequence? Does low ergothioneine cause these problems, or do the diseases cause low ergothioneine? And in fact, intervention trials suggest the ergothioneine is protective. A whole set of laboratory studies on animals and human brain organoids show it's neuroprotective. So you need that kind of evidence. You're absolutely correct. I mean, blood donors are in general very healthy. But the confounder, of course, is that unhealthy people do not or cannot donate blood, so they get filtered out. And just relying on correlation is quite dangerous because Professor David Lane, who was chief scientist at Singapore's A-Star, showed me a correlation once with R is 0.99, which is huge, between incidence of autism and consumption of organic food. And they correlate very nicely.
Must be causal.
Well, you could say, do autistic people eat more organic food, or does eating organic food cause autism? Neither is likely, but it means you've got two things that are increasing at exactly the same percentage rate. They will appear to correlate. And that's why you always need experiments, ideally clinical trials. Human clinical trials are very expensive, or you need other experiments to show that there's a causative effect. I mean, to take one example, there have been multiple studies that low levels of B vitamins or low levels of vitamin D are associated with many diseases. But studies giving B vitamins to people at risk of dementia and some recent studies with vitamin D have not shown protective effects. You've got to be very careful with these correlations. And it could well be that disease is caused low vitamin levels for some reason.
Hmm.
Some of
I
my
mean,
earliest research work was on rheumatoid arthritis, and rheumatoid subjects are often anemic. So they used to give them iron, which turned out not to be a good idea. And the anemia is a body response to the serious inflammation. You decrease body iron levels. And if you treat the RA, the body iron levels come back up. So it's not an iron deficiency. So
yeah.
beware of correlations. But you're absolutely right. I mean, people who donate blood are, on average, much healthier than people who don't.
And there are some of those experimental studies, and there are all sorts of different roads that we could go down with respect to what you just said, like how B
I know.
vitamins interact with other nutrients and things. But returning to ergothioneine, something that you mentioned is that tissue damage tends to increase local production of OCTN1. So clearly, the expression of the gene that encodes it is somewhat plastic. Do you
Yes.
think that that change in local expression is primarily driven by oxidative damage?
We're not quite sure, actually. It involves a transcription factor called RUNX1. We came across this because I had two people in my lab. One was studying ergothioneine, and the other was studying fatty liver disease. So they said, well, let's measure ergothioneine in the damaged liver. And we thought it was going to be low. It was actually high.
Hmm.
And then we found the transporter had gone up and so on. Japanese published a similar study, and then what came out was if you prevent this ergothioneine accumulation, the liver damage gets worse. So there's a lot of literature data that ergothioneine levels increase at sites of inflammation. And people have sometimes said, oh, well, you know, it's associated with Crohn's disease and so on. We think it's the exact opposite, that you pump ergothioneine in there to help protect against the tissue damage that's taking place.
Has there been any effort to try and quantify the amount of oxidative damage in different tissues and then map that to local ergothioneine levels?
Actually, no. It's very hard to do in humans because most tissues you cannot sample. It's something that needs doing because there are various biomarkers of oxidative damage, of lipid peroxidation, of DNA damage, and so on, that can easily be measured in tissues. It's one of many things for ergothioneine that still remains to be done.
Moving to ergothioneine metabolism, what's it metabolized into? And do those metabolites have important biological activities?
A major metabolite is hercinine, and in the knockout animal, you don't see hercinine, you don't see ergothioneine, so hercinine comes from it. As far as we know, and again, it's not been studied, the hercinine and there's another thing called ergothioneine sulfonate don't have direct biological effects, but they could have because nobody studied it in any detail. The metabolism seems to be pretty slow.
Finally, with respect to ergothioneine excretion, you mentioned at the start that renal resorption of ergothioneine helps us hold on to what might otherwise pass out of us. And that brings up a question related to whether patients with some sort of kidney dysfunction, say chronic kidney disease, might be much less able to hold on to ergothioneine, and therefore that might influence what's best for them in terms of intakes. Have you thought much about that? Does that make any sense?
Well, ironically, two papers have just been published from the United States showing that ergothioneine levels in patients on dialysis are extremely low. And I think they're applying for funding to do a clinical trial to see whether administering it has benefit. And I hadn't thought about that before. And then when I read the paper, I actually talked to one of the investigators. It's probably not only ergothioneine, that anything that's renally reabsorbed when the kidneys are working properly and you're just washing urea out with a great mass of physiological saline or whatever they use, is going to affect many nutrients. So I suspect dialysis patients have a series of nutrient deficiencies. And certainly they showed they had very low ergothioneine levels.
I hadn't seen that research, but that all makes a lot of sense to me. So if we move now to speak more about the epidemiology of ergothioneine and how it relates to age and to disease and dysfunction, you published a commentary on some work that came out in the American Journal of Clinical Nutrition recently. And in that, a scientist named Pierre Ayot and his colleagues found that, contrary to some other research, blood levels of ergothioneine actually rose with age.
Yes.
What do you think might explain the disparity between their findings and the more regular report, which is that blood levels tend to diminish with age?
I think, firstly, the extent of the decline with age in blood is not great in healthy people. It's much bigger in subjects with various age-related diseases. The other thing, of course, as you mentioned earlier, is the activity of the transporter may be different in different populations. The third thing is, I wasn't quite clear from that paper exactly what in their diet is contributing the most ergothioneine. So it may well be what they're eating. Again, we've studied Singaporean populations. There have been studies, I think, in China. There have been studies in Saudi Arabia. They all suggest a decline with age. Not a very big decline. So it may be different populations will show different things. That was a very interesting paper. They also mentioned this compound selenoneine, which is like ergothioneine, but with selenium instead of the sulfur. And ergothioneine hasn't been much studied, and this selenium analog has scarcely been studied at all. But in populations who eat a lot of fish, they take in quite substantial levels.
And that's taken up by the same transporter as ergothioneine, is that right? Or it can be?
There's been one in vitro paper which looked at OCTN1, and they said it also takes up the selenium analog. As far as I know, there hasn't been any in vivo confirmation of that.
That selenoneine was only discovered relatively recently. I think it was 2010. Yeah. Do you think that it might be acting in quite similar ways to ergothioneine?
It could be. I mean, in the test tube, it has strong antioxidant properties like ergothioneine. Selenium compounds do all kinds of things in the human body. So it may well have quite different chemistry. But as I say, it's scarcely been studied.
Regarding other epidemiology, there's an abundance of research showing that people with various different diseases and forms of dysfunction, everything from Alzheimer's to vascular dementia to Parkinson's to cardiovascular disease, preeclampsia, erectile dysfunction, the list goes on, have lower blood levels of ergothioneine. Are there any obvious exceptions to that general pattern? Do you think that there might be, and then they just ended up not being published because they run counter to what else is out there?
Well, I mean, publishing only positive results and ignoring negative ones is a huge problem in biomedical research. So it could well be. The interesting thing is the range of diseases where low ergothioneine correlates with, and in some cases predicts. We did a study on people who didn't have cognitive impairment but later developed it, and those who had the lowest ergothioneine levels were the ones who developed it. So it's cognition, it's eye disorders, and so on. So we've actually argued that ergothioneine is a bit like vitamin E. It's essential in the human diet, but to see the consequences of deficiencies of it may take many decades. And as you know, vitamin E was discovered as a factor needed for reproduction in rats. It's got nothing to do with reproduction in humans. Vitamin E deficiency causes neurodegeneration, but the only time you see that is if there's a defect in the proteins that handle vitamin E in the body, or if there are defects in fat metabolism, because it's a fat-soluble vitamin. So we have a feeling ergothioneine is the same. The late Professor Bruce Ames called it a longevity vitamin. So it's something that you need but you don't see the consequences of not having enough until later on in life. He argued that there are many of these. I think he also mentioned taurine as one.
He did. Yeah, taurine, a bunch of others. Cuine was one of them, which was particularly obscure, and there's still not been much research on that. But that paper was certainly very prescient, I think, and that's how I first came across ergothioneine, actually.
Okay, yeah.
Moving on to experimental studies. If we go back to your proposal to include ergothioneine in the ITP, then there has been some work looking at effects on lifespan in male mice already, showing lifespan extension and a bunch of other positive effects, namely slowing of age-related changes in function and physiology. What did you make of that study? I suppose that you must have found it somewhat compelling to put forward ergothioneine for the ITP.
We looked at some of the published data, and the lifespan extension looked quite impressive. So I thought it was important to have a very rigorous, I'm not saying that paper wasn't rigorous, but to have a very rigorous controlled conditions which ITP delivers. So it'd be very interesting. Of course, more important than lifespan is health span. Will it protect you against age-related diseases? Which is really a very important issue.
Some of the early work on that has really focused on the brain, like your pilot work that you mentioned earlier.
Yes.
And some other research that used a lower dose. I think the other publication on that subject gave people just 5 milligrams a day and found that the increase in blood ergothioneine was correlated with improvements in memory in the participants in that study. So that's certainly promising. And then going back to what you were alluding to earlier and trying to tie some threads together, it seems like ergothioneine might be really important to the development of the brain and possibly to development in general. So if it's crossing the placenta and if it is in breast cells, is present in breast milk, I believe that the expression of OCTN1 in the breast actually increases during lactation too. And then it's possibly affecting the brain of the newborn. Then one of the subjects that I just thought this might be relevant to is concussion, different types of head injury, where you have that type of trauma to the brain. And you mentioned that various forms of damage can acutely increase OCTN1. What do you think about ergothioneine as a potential nutraceutical intervention during concussion and other forms of brain injury?
Well, one of my colleagues in the anatomy department at National University of Singapore, Professor Ong Wei, has worked with colleagues in Taiwan, and they've looked at ergothioneine in various stroke models, and it's very neuroprotective. Interestingly, in a lot of these stroke models, most compounds only protect if you give them before the injury, which of course doesn't help you when you're dealing with human stroke. But they've shown that it has some degree of protection after the injury. Another study that we did, and has now been picked up by Professor Chester Drum in our Department of Medicine at NUS, is heart toxicity. There are certain compounds used in chemotherapy that are very effective anti-cancer drugs, but they damage the heart. And that damage has been widely thought to be due to too many free radicals. And in both our studies, ergothioneine has been extremely protective against cardiac damage. That's another area that... And of course, the other thing about ergothioneine is that our studies and many others indicate that it's extremely safe. So if you can make a scientific case for doing a clinical trial, you can go straight into the clinical trial. All the toxicology and so on has been done. And I know there are clinical trials being planned for kidney dialysis. Of course, we're looking for funding, waiting to hear the outcome, to redo our clinical trial, but with a much broader range of biomarkers of neurodegeneration. I know Professor Douglas Kell in the UK is trying to pursue the preeclampsia effect, because what he's shown is that women at risk have low ergothioneine. And of course, preeclampsia is a devastating disease with often bad consequences for the mother and the child. So I hope he gets the money.
you mentioned chemotherapy. I think there's also some work suggesting that it can help preserve sperm numbers and sperm morphology or function during exposure to some chemotherapy drugs.
There have been one or two studies on this. One very interesting thing that, as I mentioned, ergothioneine is present in all body fluids, in plasma, tear fluid, saliva, wherever you look. It's also present in seminal fluid, including human seminal fluid. But the seminal fluids of boars and stallions have an enormous activity of ergothioneine. And there have been one or two papers of feeding horses ergothioneine to see if it makes them run faster. But they've measured various physiological parameters, but they didn't mention running speed and endurance. So again, that's an interesting comparative physiology, like why do boars and horses have such high seminal fluid ergothioneine levels?
And are there any other applications that you find interesting? I know there's been some work on the respiratory system too, and protecting the lungs from certain stresses.
Very little work. There's a group in Scandinavia that's been looking at ergothioneine levels in children. And they say that the higher the levels, the lower the risk of various allergies and so on, which goes back maybe to developmental issues that you mentioned. We have a very good cohort in Singapore called Gusto, which has been following pregnant women, then the children since birth, and looking at all kinds of things that influence children's outcomes. So I'm trying to work with them. They're very interested in determinants of children's outcomes. So that may be a whole area for ergothioneine. But again, you have to be very careful, because high ergothioneine level in the baby means high ergothioneine level in the mother. It goes from mother to baby. And of course, breastfeeding and healthy mothers tend to give you healthy children. So again, it doesn't mean the link with high ergothioneine and low risk of disease doesn't mean a cause-consequence thing. It needs experiments.
One subject related to that I think is very interesting is the fact that mothers tend to experience a lot of sleep and possibly circadian rhythm disruption too when they have a newborn. And I know there are some people who are starting to look at ergothioneine and its effects on sleep. So I wonder if there could be some
interesting Japanese studies that ergothioneine promotes healthy sleep. The study in the US that looked at people with self-diagnosed cognitive impairment, the worried well, they found ergothioneine was beneficial. They also reported sleep improvements. In fact, we've just got a grant to study this. We have a very good sleep laboratory at NUS, Professor Michael Chi and Dr. Helen Zhao. And the effects on sleep seem to be demonstrated based on the Japanese and the American studies after quite short periods, like 8 weeks, 16 weeks, which means you can do quite a quick study. So we just got funding to do a placebo-controlled study.
Oh, great.
On people who we know have sleep issues. And of course, as you know, many elderly people have sleep problems, and sleep disruption increases your risk of dementia, and dementia gives you sleep problems. So again, if ergothioneine improves sleep, that could be yet another mechanism. But our study will hopefully pin it down one way or the other.
And this conversation falls under the umbrella of how ergothioneine intake interacts with lifestyle. And one subject that within that umbrella is very intriguing, in part because of research on other nutraceuticals, is how ergothioneine might interact with exercise. I mention other nutraceuticals because some of the other candidate geroprotectors out there, such as metformin, seem to negatively affect how people respond to exercise training. And I suspect that might be true of rapamycin as well, and how it interacts with some types of exercise, like resistance training. But based on what we've seen so far in preclinical models, it seems that if anything, ergothioneine tends to improve muscle mass and exercise performance, going by Spiegelman's work and some of the other research. Do you think that's likely to be the case, or do you think that we really are going to struggle to extrapolate the work that's been done on rodents to humans?
Well, I think nutritionists don't know a great deal about human nutrition, but I think one thing everybody's agreed on is exercise is good for you and probably increases health span. There is a limit. People argue, do you need 5,000 or 8,000 or 12,000 steps a day? Running marathons probably not good for you in the long term, but moderate exercise seems very good. Having a diet rich in fruits, vegetables, grains seems to be good for you. And of course, that diet will also be rich in ergothioneine, especially if there's a lot of mushrooms there. Whether there's an interaction, the animal studies strongly suggest it, but you can't beat human studies. So I would encourage people to do that.
Mm-hmm. Regarding interactions with other dietary constituents, I read in one of your review articles that vitamin D might promote the expression of OCTN1, which would be an interesting example of those interactions. Do you think that there's anything that might synergize with ergothioneine?
That was, if I remember correctly, a rat or a mouse study, and they were using a lot of vitamin D. So, I mean, this is a key question of how you interact with other nutrients. We're unaware of other nutrients that compete with the transporter, but the other nutrients might regulate transporter activity, and that's really a field that's scarcely been studied. So, and then, of course, the other question is if we combine ergothioneine with other products in supplements, do we get a synergistic benefit? And that's something we are studying. Now, doing full double-blind placebo-controlled clinical trials on every conceivable supplement mixture you can get is completely impossible, but there are shortcuts that can give you indications. And of course, there are a number of things on the market now that are supplements, including ergothioneine. My only caution is with the nicotinamide riboside and the B vitamins and melatonin is think about quality control of exactly what is in the supplement.
If I had to pan back and try and come up with some sort of simplistic conceptualization of what ergothioneine does, then I think part of that would be that it seems to step in and take up some of the slack when the body's under a lot of duress. And maybe that could be true of when, for example, the levels of other antioxidants are insufficient. Do you think that that could be true? Do you think it might interact with some of those other antioxidant systems and vitamins, for instance, in that way?
What intrigued us was this evidence that ergothioneine is accumulated by raising OCTN1 levels at sites of injury, because we don't see that. I did a literature search. We don't see that with other antioxidants. So we don't seem to raise vitamin E or vitamin E levels in tissues as a response to injury. If anything, they tend to decrease. Now, of course, that doesn't mean that the ergothioneine is acting as an antioxidant. But I mean, for example, in a lot of diseases, glutathione levels drop. Normally, there's a lot more glutathione than ergothioneine in cells, but maybe in tissue injury, the glutathione goes down and you bring in more ergothioneine. But of course, as I mentioned earlier, ergothioneine seems to be capable of doing a lot of things.
Finally, regarding interactions, do we know anything about how it might interact with any medications? Are there any contraindications there?
We have not seen anything so far. And ergothioneine metabolism hasn't been well studied. And in fact, as you know, a lot of nutrients and a lot of drugs, the initial metabolism is in the gut itself, which is why drinking grapefruit juice, which contains inhibitors of some of the P450 enzymes in the gut, can in fact actually increase your dose of a number of medications.
Yeah, a lot.
By blocking this can be very clinically significant with a number of drugs. We haven't seen anything yet, But then again, we haven't really studied ergothioneine metabolism.
And so when it comes to recommendations, I think the approach that a lot of people with sufficient resources tend to take to this type of thing is to try and look at some biomarker of intake or of activity and then dose things according to some of their test results. And in your excellent annual reviews paper, you very tentatively suggested that a plasma level of over 810 nanomoles of ergothioneine seemed to generally be associated with health. But it's now a few years later in that paper. Would you stand by that as a, again, emphasizing the word tentative recommendation?
I would stand by it, but just to point out that all the studies were done on the Singaporean population. There's been a very recent paper from Japan on ergothioneine and risk of dementia, which came out with broadly similar levels. So that seems to be the level that if you're at or below that, your risk of age-related diseases is significantly increased. Is that the optimum level? I think I mentioned earlier the case of vitamin C. If you're below about 20, 30 milligrams a day, there's an increased risk of scurvy. But is that the optimum level, or do you need more? I think that's the question people ask with all nutrients. And with vitamin C, maybe you need 100 milligrams a day. If you smoke cigarettes, you definitely need more. But on the other hand, if you start taking 2 grams a day, then you begin to see possible toxicities.
I wonder if over time we'll see that some of those polymorphisms in the gene that encodes OCTN1 might relate to certain historical exposures. So take the example of the quantities of mushrooms that generations of people have consumed. Or maybe another one could be something like the altitude at which people live. But another question that comes up is whether the optimal dose might relate to people's bodily levels. So would you expect that someone with quite low bodily stores of ergothioneine would respond better to supplementation than someone who already has quite high levels?
In fact, in our clinical studies, we will be screening people and pick out the ones with low ergothioneine. And for priority inclusion in the clinical trial. But we don't want to just include people with low ergothioneine. We want a range of people. But the starting level will be extremely important. And in fact, that's been shown with vitamin E and vitamin C, where people have looked at their effects on biomarkers of oxidative damage. And quite often they found no effect. And it turns out that if your levels are low to begin with, you're more likely to see an effect. And of course, the people who volunteer for clinical trials are often healthier than the average, so to speak. So certainly they may have higher levels of nutrient. So if people ask me, should I take ergothioneine supplements? I said, well, look at the evidence. Try and ensure that you get a product from a reputable manufacturer with quality control. But you might also consider just eating more mushrooms.
Now, I realize this is speculation, but regarding daily intakes, do you have any sense about what the sweet spot might be for a typical adult?
We don't know, and this is something we're actually working on. And it goes back to the question of why do some people have low levels? So
when thinking about ergothioneine intakes last week, I saw some parallels with creatine monohydrate, another dietary supplement. Creatine, like ergothioneine, accumulates in some tissues, and it can take several weeks to fully saturate creatine stores, much the way that seems to be true of ergothioneine stores. And one approach that gets taken with creatine supplementation is to use a so-called loading dose. Whereas if you take a consistent dose every day, let's say you take 5 grams every day, you'll probably after several weeks fully saturate various stores. Certainly that's true of skeletal muscle. Whether it's true of the brain, we're not sure. I thought, well, one approach with ergothioneine could be to initially take a much higher dose to try and saturate stores. And thereafter, one could take a maintenance dose, which might just be something like 5 milligrams. Do you think that's a logical approach? Do you think that by doing that, you would reach the same levels that you would ultimately get to were you to just use the same lower dose day in, day out?
Well, I think the simple answer is I don't know because we've never really done those experiments. My instinct would be to give fairly low levels or eat more mushrooms because it is very quickly accumulated. Interestingly, we've never found any subject with no ergothioneine. Um, and of course, in the knockout animals, there is no ergothioneine. And maybe that means that the transporter is essential. So that if you had a mutation that deleted it, you don't survive. But that's pure speculation. So,
regarding the source,
creatine,
then,
I don't know, but to be honest. But I know that there's evidence that it improves muscle function. There's some evidence it may help frailty and that kind of thing. So, and it does take a while to accumulate. But of course, creatine will raise creatinine, which but doesn't seem to be a problem in most people. But we never worked on creatine. But that's just based on things that I've read.
with respect to mushroom consumption, you just encourage people to try and consume plenty of mushrooms on a regular basis.
Mushrooms seem to convey health benefits by several mechanisms. And we think ergothioneine is a very important one. But they also contain polysaccharides and all kinds of things that people have worked on. It's like this question, should we combine ergothioneine with other nutrients? Or is it a good compound by itself? And we need a lot more research on this area.
Yeah, and it parallels discussions of psychedelics too, because I know some people speak about how if you take an equivalent dose of psilocybin from magic mushrooms, then the experience is different than the synthetic dose because of the so-called entourage effect. There are other tryptamines that are having some effects on the experience, and perhaps that's true of ergothioneine and some of those other substances in mushrooms too.
It could be. I mean,
greater benefits from the mushrooms.
What we know is that diets rich in fruits, vegetables, grains are associated with improved health, an improved health span. Nobody has, as far as I know, found any single component that accounts for that. So people have said, oh, it's the antioxidants, and then they feed people lots of antioxidants. Nothing much happens. Maybe it's the fiber. Well, again, fiber has benefits, but it cannot replace the whole effect. Maybe it's the sulforaphane. Again, I think you need this combination.
And then finally, there's the question of intake timing. I know the levels of some endogenous antioxidants vary with time of day, like glutathione, like peroxyredoxins. And I also know there's been some work on other animals suggesting that blood levels of ergothioneine are higher during the daytime or the active period, more specifically, because a lot of this research is on nocturnal animals, than during the sleep period. And that raises the question about whether there is a best time of day at which to take them, going back to what we were discussing with respect to intestinal uptake too. But being pragmatic, people probably don't need to worry about that too much. But maybe there'll be a point where we find that it does actually interact with the circadian clock in some interesting ways.
Oh, I would not at all be surprised if it does. I think it's a molecule with many profound effects. But I mean, my simple answer is I don't know because nobody's done the studies.